Hello, this is Mr. Marriott with Noteworthy Science. Today we're going to be learning the additional higher level component of subtopic B3.1 Gas Exchange. Under the curriculum matrix, this falls under theme B, form and function, and the level of organization of 3, organisms. The guiding question that is relevant to the additional higher level component is the first one. How are multicellular organisms adapted to carry out gas exchange, specifically that of humans? In the additional higher level learning outcomes, there are 3 new ideas that we're going to explore. Now, unlike the SL component that looked at both animals and plants, this one only deals with that of humans. And specifically, how is hemoglobin adapted to really carry out its function? And while hemoglobin has been explored in other subtopics in this course, this subtopic really starts to dive into questions that are really interesting, such as, if hemoglobin is so good at binding oxygen, why does it ever let go? What is the interplay between oxygen exchange between a pregnant woman and her fetus? How exactly does gas exchange work in this scenario? So in today's subtopic, we'll be exploring all of these questions. Let's begin. This topic is all about hemoglobin, and specifically what makes hemoglobin such a good protein at both taking oxygen in areas like the lung, but also offloading oxygen where it's needed. So before we begin, I think it's a good idea to review what makes an erythrocyte, or red blood cell, so good at holding hemoglobin and at delivering oxygen. So the main question would be, what adaptations do erythrocytes have? How is their form related to their function? So erythrocytes are somewhat unique in the human body in that they are anucleated, meaning that they don't have a nucleus. Now this is quite odd, especially when we consider things like cell theory, as we discussed in A2.2. But it means that they have even more room for hemoglobin. So this affords them more space to pack even more hemoglobin in. Concavity way is to take a cross section, and you can really see its shape here. So its shape here is something called biconcavity, or it has a biconcave shape, bi for 2. So it has a double concave shape here, so it's almost like a figure 8 shape. Now, why does that matter? If you were to compare that to this shape, this is a far greater surface area to volume ratio than this one. So this really promotes gas exchange. So red blood cells are really highly adapted at holding hemoglobin. So what's hemoglobin again? Well, hemoglobin is a quaternary conjugated protein. Now, let's recall what that means. We looked in the protein subtopic, we studied this in more detail. Quaternary means that it's not composed of a single polypeptide, but more than one. In this case, hemoglobin is composed of 4 polypeptides, 2 alpha chains and 2 beta chains. However, I also said that it's not just a quaternary protein, it's a conjugated protein. So what do I mean when I say conjugated? Well, conjugated means that it's not just comprised of polypeptides, it also comprises a prosthetic or a non-protein group. And in this case, it's its namesake. It also contains a heme group. So what is a heme group? A heme group is a prosthetic group that contains the atom iron. And it's this iron that gives its red coloration and what makes your blood red in the first place. This iron is able to bind to an oxygen molecule. It actually binds at roughly a 120-degree angle, but this is where the magic happens, so to speak. At every single heme group, it is able to bind an oxygen molecule. So because there are 4 heme groups, that means at maximum, one hemoglobin protein can bind a maximum of 4 oxygen molecules. Let's take a look at a graph, something called an oxygen dissociation curve. So let's draw a graph right here. And on the x-axis will be the PO2, which stands for the partial pressure of oxygen. Now this might seem like an odd unit to use. Partial pressure of oxygen is a pressure measurement, therefore this will be in kilopascals, or you may also see this in millimeters of mercury. Either way, this is a pressure measure. So what kind of pressure am I talking about? Well, the air I'm breathing right now is roughly 20% oxygen. So what the partial pressure of oxygen really means is the pressure of oxygen if it alone occupied the entire volume of this air. So it's another way of saying how much is oxygen contributing to the air around it. So the higher the partial pressure of oxygen, basically the more oxygen there is. Now on the y-axis is going to be the percent saturation of oxyhemoglobin. Basically, what percentage of hemoglobin is bound to oxygen. So at 100% saturation, that means every single hemoglobin molecule is bound to an oxygen molecule, or 4 oxygen molecules. At 0%, it means no hemoglobin molecules are bound to oxygen. So what is the relationship between the partial pressure of oxygen and the percent saturation of hemoglobin that's bound to oxygen, or oxyhemoglobin? So what you might have predicted is a positive relationship. Let's imagine that it is a linear relationship. This seems intuitive. This means that the more oxygen there is, the more hemoglobin is going to be saturated with oxygen. That kind of makes sense. But hemoglobin is a lot cooler than that, because the relationship, while it is positive, it is not linear. It is what's called sigmoidal, or S-shaped, where instead of a linear function, we get something that looks like this. Now, this is quite important. So a linear relationship implies that the more oxygen there is, the more saturation it is at any given pressure. But what a sigmoidal curve means is something slightly different. What this curve means is that at a very low amount of partial pressure of oxygen, there's a very small rise, meaning that it's actually quite difficult for hemoglobin to bind onto oxygen. But at some critical point, you see that at a very narrow band here, a very small increase in partial pressure of oxygen has a very large increase in the percent saturation. And then once we approach 100, it starts to level off again. This is really, really important. This means that effectively, it's hard for hemoglobin to bind to oxygen at first, but then it becomes really fast at doing it, and vice versa. It's really hard to get rid of oxygen at first, but once it happens, it offloads very quickly. The reason for this relationship is the cooperative binding nature of hemoglobin. So what I've drawn here are the 4 different sites of oxygen binding. So here are 4 heme groups. And what I've shown here are 4 different binding sites. Now, when there is hemoglobin in a not oxygen bound state, notice that the binding sites I drew them quite small. This is to represent that it's actually quite difficult for it to bind oxygen in this state. Now, in a really high concentration of oxygen, the likelihood of it binding to oxygen is going to go up. So let's imagine an oxygen molecule finally binds. Once this happens, it induces a conformational change in the shape of hemoglobin. Specifically, it moves a little bit, causing the subsequent or the adjacent section to change. And what happens is that I've drawn here, the binding becomes actually easier. So another oxygen molecule now is going to bind much easier, and that in turn is going to do the same thing. It's going to cause a conformational change on its adjacent chain, and so on, until finally 4 different oxygen molecules are all bound. What this is saying is that as each oxygen molecule binds, it promotes the binding of every subsequent oxygen molecule. So you might refer to this as a cascade of oxygen binding. But the major process or major term that we're talking about here is something called the cooperative nature of oxygen binding. Now, this is really, really adaptive. So this means that the two most probable states of hemoglobin is its deoxy or T state for tense state, and its oxy or R state or relaxed state. So these are kind of the two most probable ways, because with every oxygen binding, the next becomes progressively easier, making this very likely. But the moment an oxygen gets offloaded, offloading of the rest becomes very easy as well. So why does all of this matter? Well, this is actually very, very highly adaptive. Specifically, hemoglobin is going to have a much higher affinity for oxygen in really oxygen-rich areas, like the lung. What that means is that in areas of very high oxygen, it's going to promote oxygen loading, meaning oxygen loading is going to happen in areas where oxygen is really high. And this is also true in the reverse. Hemoglobin will have a very low oxygen affinity in very oxygen poor areas, such as tissues that are undergoing high amounts of aerobic respiration, such as the muscles. So that means in those areas, oxygen unloading is going to be promoted. So in this way, oxygen is being able to be taken where it's very high and being able to be delivered and used where it's really, really needed. How does all of this fit in to a woman who is pregnant trying to nurture her fetus? Because both the fetus and the mother require oxygen. So we have a little bit of a problem here. If the fetus also used the same hemoglobin as the mother, it's almost as if the two proteins are in competition with each other. And so the mother might be not able to deliver as much oxygen to her fetus. So there's actually a very cool evolutionary adaptation around this problem, and that is fetus does not use the same hemoglobin as its mother. So let's just do a quick recap of this scenario here. And this was covered in the reproduction subtopic. So a mother would be inhaling, having gas exchange in her lungs. Oxygen is going to be carried by her blood vessels to the placenta. Oxygen is going to diffuse into the placenta and then be delivered to the fetus via the umbilical cord. The reverse is true with carbon dioxide, which is going to be provided by the fetus, diffusing into the placenta, and finally towards the mother. So the question is, how is the fetus able to acquire oxygen in spite of the fact that the mother's also going to require oxygen? Well, as mentioned, the mother uses what we call adult hemoglobin. And this is the hemoglobin that all of us have. Adult is kind of a misnomer because it's not just when you're an adult that you have this, it's actually within a few months after being born. But a fetus is going to have fetal hemoglobin. So let's see how these two types of hemoglobin differ from each other. So adult hemoglobin, as we've already discussed, is composed of 2 alpha subunits and 2 beta subunits, able to bind to 4 oxygen molecules. So how does fetal hemoglobin differ? Well, fetal hemoglobin is made up of the same 2 alpha subunits, but instead 2 gamma subunits. It's a very similar structure, but this key difference between the gamma subunits and the beta subunits is all the difference in the world. So to understand why this matters, I want to bring your attention to a molecule called BPG, or 2,3-biphosphoglycerate. Now, this molecule, which is present in the blood, actually promotes oxygen offloading. So when 2,3-BPG is around hemoglobin, what happens is this molecule is able to bind within hemoglobin. Specifically, it binds quite well to both beta chains. When this occurs, it causes a slight conformational change, making it more likely that oxygen gets offloaded. So in areas of higher 2,3-BPG, you're going to get more oxygen offloading. Why does this matter? Well, 2,3-BPG cannot readily bind to fetal hemoglobin. And that's because rather than beta subunits, there's gamma subunits. This means that in the presence of 2,3-BPG, if there is both adult hemoglobin from the mother, but also fetal hemoglobin, it will promote oxygen offloading in this hemoglobin, but not this one. Therefore, fetal hemoglobin actually has a higher affinity for oxygen at the same partial pressure. Let's do a prediction here. If this was our original graph, so let's say this is for adult hemoglobin, predict what a curve would look like for fetal hemoglobin. So fetal hemoglobin, as we mentioned, has a higher affinity for oxygen at every partial pressure of oxygen. So in effect, it's like you're taking the curve and you're shifting it to the left. So you might see a curve that looks something like this. So this curve right here is something called the oxygen dissociation curve. And it plots this oxygen saturation over different partial pressures of oxygen. So as we've just predicted, fetal hemoglobin has a higher affinity for oxygen at every single partial pressure of oxygen. Let's take a look at the percent saturation hemoglobin at roughly 50%. Notice that in this case, that amount was reached at a much lower amount of partial pressure of oxygen for fetal hemoglobin than it was at adult hemoglobin. Or another way of looking at this was, let's take a specific point here. Let's say this point right here. If we draw a line up from this point, then that means at any given partial pressure of oxygen, the fetal hemoglobin has a higher affinity than the adult hemoglobin. Therefore, fetal hemoglobin is able to acquire oxygen much more readily than adult hemoglobin at the same partial pressure. This is really, really adaptive and really, really important for fetuses. Now, the question might be asked, why then switch to adult hemoglobin if fetal hemoglobin is so good? Well, the difference is that fetal hemoglobin is really good at acquiring oxygen, but adult hemoglobin is actually better at unloading it. So as an adult with a higher demand, adult hemoglobin is actually more adaptive than fetal hemoglobin. So fetal hemoglobin is extremely important in a very early stage during pregnancy and throughout pregnancy, but the moment that that organism is now self-sufficient, so to speak, and not relying on the mother, its demands have to be met by adult hemoglobin in a much better capacity than before. The last thing that I want to talk about is the role of carbon dioxide. I think a pretty big misconception about hemoglobin is that it only is associated with the transport and binding of oxygen. But actually this is not true. There's another really big important respiratory gas that we haven't talked about, and that is carbon dioxide. So how does all of this work together with carbon dioxide? Carbon dioxide is going to be produced as a byproduct of aerobic respiration. So what happens is that when aerobically respiring tissues produce carbon dioxide, they release that into the blood. As they release that into the blood, it's going to go into the plasma, and then that carbon dioxide is actually going to diffuse within an erythrocyte. Now, a few things can happen here. One thing that you may not have been aware of is carbon dioxide can actually bind to hemoglobin directly, something called carbaminohemoglobin. Something else that could happen though is carbon dioxide, those of you studying chemistry should be well aware of, is carbon dioxide in the presence of water can form bicarbonate and carbonic acid. And once that happens, you start to form hydrogen ions. As hydrogen ions are formed, this reduces the pH. If you reduce the pH, this actually decreases hemoglobin's affinity for oxygen because it actually induces a conformational change, making oxygen binding less likely. So taken all together, why is this actually adaptive? Well, this is adaptive because any oxygen is going to be released and delivered wherever carbon dioxide is really, really high. In effect, any area that has high carbon dioxide are going to be tissues that are respiring a lot and thus are going to need a lot of oxygen. Therefore, any area that has high CO2 is going to have high oxygen offloading. So let's again take a look at our oxygen dissociation curve and take a look at the role of carbon dioxide. What's happening with carbon dioxide is something called the Bohr shift. Now, those of you who have again studied chemistry might be like, oh, Bohr? Like Niels Bohr? Actually, this is a different Bohr. This is Christian Bohr. And this curve was named after his discovery of it. And so here again is my original curve in red. But instead, as we saw with fetal hemoglobin, the affinity of fetal hemoglobin was higher than that of adult hemoglobin. Thus, it was left shifted. However, the Bohr shift is a right shift. Why? Well, in areas of high carbon dioxide, there's going to be a lower affinity for oxygen. So that means at any partial pressure of oxygen, the affinity with high CO2 is going to be less. Now, this is really, really important because where is the partial pressure of oxygen going to be low? In respiring cells. Wherever there's respiring cells, there's going to be a lot of carbon dioxide. Thus, lower pH, conformational change, lower oxygen binding. Thus, the affinity is going to be lower. Thus, there's going to be a lot of oxygen offloading. This is not so much a factor in the lungs because the partial pressure of oxygen is going to be so high that this shift is effectively irrelevant. So what the Bohr shift is allowing to happen is a promotion in oxygen offloading in areas that need it. So the idea is that an increase in carbon dioxide causes an increase in oxygen dissociation, meaning offloading. So taken all together, what have we looked at here? I know there was quite a bit. So to recap, hemoglobin is a protein that binds oxygen. Its major role is to acquire oxygen in the lungs, deliver it to tissues, and offload it to tissues that require it. And a few ways that this is adaptive, the major one is the fact that it binds cooperatively, meaning that it's hard at first for oxygen to bind, and then it becomes subsequently easier with each oxygen that is bound to it. The adaptation in fetuses is that their version of hemoglobin, fetal hemoglobin, has a higher affinity for oxygen than that of their mother, allowing them to acquire the necessary oxygen for their growing tissues. And finally, carbon dioxide enables what's called the Bohr shift, whereby any tissues that have very high amounts of carbon dioxide because of their high rates of aerobic respiration are going to be areas where hemoglobin is going to be more likely to release and offload oxygen. So taken all together, hopefully the takeaway is, man, hemoglobin is a much cooler molecule than I ever gave it credit for, and how much adaptation is happening at such a minute level for me is really just mind-blowing. Let's review the additional higher level learning outcomes from subtopic B3.1 Gas Exchange. While there are only 3 learning outcomes in this subtopic, these are moderately complex, and we're really looking at the interplay as to how the protein hemoglobin is so good at its role. So the first learning outcome is looking at how specifically does fetal hemoglobin and adult hemoglobin differ, and how both of them are so well adapted to gas exchange. Specifically, how are they really, really well at picking up oxygen, and what interplay is there between the fetus and the mother during pregnancy? Really understanding how hemoglobin works has all to do with the cooperative binding nature of oxygen, and this really is tied to all the other learning outcomes. Specifically, understanding the cooperative binding of oxygen helps explain both the Bohr shift and understand the S-shaped or sigmoidal curve that's actually going on when this is happening. So the Bohr shift all has to do with carbon dioxide. What role does carbon dioxide have in actually lowering oxygen affinity within hemoglobin, and why this is so important for delivering oxygen to tissues that need it? And this final learning outcome here really is understanding how to read the oxygen dissociation curve. So these three learning outcomes are kind of all related to each other, but I believe the strongest core linking all of them is the cooperative binding nature of hemoglobin and how this S-shaped curve can differ both in fetal hemoglobin and in carbon dioxide concentrations. If you wish to learn more, which I always encourage you to do, this last page is a good place to start. These textbook pages are those that I've consulted in developing the entirety of B3.1 content for my notes. The 3D models here are really useful in really delving deeper into some complex structures. Specifically in this subtopic, we really looked at hemoglobin, both adult and fetal hemoglobin, and these articles from the PDB 101 really help explore how that conformational change occurs when oxygen is binding and when 3-PBG is also binding as well. The rest of these models and simulators really have to do with the standard level content, but it is also worth going into just to review. This article is of particular interest because it talks about how CRISPR can be used in order to unsilence the fetal hemoglobin gene and how this can be used to treat sickle cell anemia. This is a concept that we've also looked at in a previous subtopic. This article right here is just really fascinating because every time that you talk about lungs, you're going to see a stat thrown around such as the human lungs are roughly the equivalent of how the surface area of a tennis court. And this paper is really talking about, is that really true, yes or no? It's a really fascinating read. At the end of the day, what I would really encourage students to do is not just learn for the sake of a test, but really just learn for the sake of learning, learn for enjoyment.